Alright guys, here today we're going to do the first video in chapter 14, which is all about Mendelian genetics. Up until this point, if you've been following along with the PowerPoint has kind of been skipped about 25 slides. So yes, we've skipped a lot of information that I never did a video on, but that's okay. A lot of that information should be basic review. We've done an activity in class that helps you with that review, and I kind of want to pick up where some of the stuff that we're going to get into is either brand new or is much more interesting, and that's the kind of stuff you might actually enjoy listening to on these videos. So that's what we're going to get going on to here. Now, the information that we talked about was all of, that we should have done in review prior to this video, dealt with all of the concepts that Mendel Gregor Mendel had come up with his pea plant experiments that described how organisms pass their genes on to the next generation and how those genes are ultimately expressed in what we see as to be called the phenotype. Now, the stuff we're going to get into today deals with predicting how those organisms that are being born, the babies that get these genes, predicting how they're going to look. Okay, and so some of this, again, I'm going to skip because a lot of this stuff you guys are great at, and so I'm going to skip the Punnett square stuff. Okay, I'll provide some resources for practice, but classically I skip this because it's one of those things that gets hammered really, really hard in Bio 1, and we may do some brief review on it in class, but I'm going to skip it for purposes of this video. Okay, so here is the slide that describes setting up a Punnett square and what that entails, and this kind of gives you an idea of how to set one up. If you needed a little refresher, you can kind of look at it, and probably just looking at this will give you some insight as to what you're supposed to do. And here is the setup of a dihybrid Punnett square, where you track the inheritance of two traits together in the babies given by the parents. And so again, I'm not going to go into how to do these. If you'd like some help, you can set up a personal meeting on those Wednesday classes that we have, or just anytime. I'd be glad to go through a crash course on how to do this, but I don't think we need to waste any class time in describing that. Okay, so monohybrid Punnett squares, dihybrid Punnett squares, you should have a good enough handle on those things that we can kind of skip them right now. I'm not saying that they're not important, because they are, because they can help you get some of these genetics problems correct that you might have to be working on. But what I do want to get into is a discussion of something known as the laws of probability. Okay, and we can utilize the laws of probability, and if you've taken probability and stats, you probably have heard of some of these, though that teacher may cover them a little bit more in depth than I do. But you can utilize the laws of probability to also predict what an offspring of two individuals might look like if you know those individuals' genotypes. Okay? And so I'm going to talk about how to do that here, and then the next time we see each other in class, we're going to work through some problems in class that are going to be daunting if you use Punnett squares. Very daunting if you use Punnett squares. And really the only ways to do these problems is to use the rules of probability. Okay? Now, Mendel's laws that we had talked about, specifically the laws of segregation, and also the one we had talked about in chapter 13 a little bit, the law of independent assortment, reflects the rules of probability and then kind of takes those laws and then applies the rules of probability to genetics problems. And so those two laws together kind of describe the fact that the alleles of one gene segregate into gametes independently of another gene's alleles, and we can utilize that idea to set up genetics problems using the laws of probability. Now, the laws of probability, if you have not heard them, there are two of them that at least that we're going to use. And so the first one is called the probability rule of multiplication. When I was in college, my professor called this the AND rule, and so I've always called it the AND rule. And so this describes the odds of two or more independent events occurring together, and that's the key here, that two events are occurring together. So in other words, you can say, I want to know what the probability of this and this occurring together is, okay? So the odds of 2 or more independent events occurring together is the product of each of those independent events. So that'll make more sense when we see the genetics problem set up in the next slide. So let's go ahead and put a genetics problem up here, a very simple one that could be solved with the use of a monohybrid Punnett square. Okay, and in the next slide you'll actually see one of those. But I want to approach it using this probability rule of multiplication and kind of at the same time use that monohybrid Punnett square to help solidify this idea in your mind. Okay, so the question states, in a cross between pea plants that are heterozygous for flower color, meaning that they have a big R allele for flower color and a small r allele, what is the probability that the offspring will be homozygous recessive? Okay, so we want to know what is the probability that this offspring is going to have 2 little r's. That's what it's telling us here. When both of the parents are heterozygous for that flower color. All right, so let's test it out. Let's look at it. Now, I've put here, I haven't put here, I should give credit to the people who actually did this at the book company, but what the people at the book company have done is they have used pennies for this graphic, and that illustrates the random nature of random fertilization and the fact that we don't know which allele we're going to get when our parents conceived us. It's random, okay? And so it's kind of like a coin flip when you're talking about 2 different versions of an allele, which we actually see here, okay? So I'm gonna mark up this document here pretty good. So, oh man, I hit the wrong one. Let's do this one. All right, so I'm going to mark this document up pretty good here. And I want to explain what you're seeing in front of you. Okay, so if we, let's see if I can do that. I don't know if I want to do that. Yeah, I don't know if I want to do that. But what we see here is the genetics from the father plant. Okay, and I wish these were kind of... well, I'll just do this. It's got a big R there and a small r there. Okay? So the father plant is heterozygous. That's what the problem states. And so is the mother plant. Okay? Now, when those particular plants, the mother and the father, go to make their gametes, the sperm and the egg, if each one of them, if they are this in their normal diploid cells, okay, when they go to make gametes, what's going to happen is that these versions of the alleles are going to segregate. That's what this is, the law of segregation. They are going to segregate into separate gametes. Okay, so if a parent's normal diploid cell is heterozygous, the big R will separate into its own sperm, and the little r will separate into its own sperm during meiosis. Okay, so this is what we get. Now, when we do this Punnett square, what we have to take into account now are the odds of these things happening. Okay, and that's what we have listed next to us. And when we use the laws of probability, we have to take into account the odds of these things happening. Okay, now we have to think about it this way. To make this baby right here, let me change the color. We'll go with this orangish red. When we try to make this baby, when the plants try to make this baby right here, we know that a big R, we know that we need a big R and a big R. Okay? So we have to think to ourselves, okay, what are the different probabilities that Dad is going to give a big R or a little r? Okay? So in this case, we aren't sure if this sperm right here is going to be a big R or a little r. We don't have any certainty, but we have a probability. the probability that this sperm is going to have a big R in it is 1/2 or 50%. Okay? And so we can say, okay, if we get that 50% chance actually happens, then the big R from dad will be represented right here in the baby. Now, we have to think about mom as well. What's the chances that mom is actually going to give her big R? Okay, because she could give a big R or a little r. We don't know that. Based on random fertilization, the fact that we can't really pick out which eggs are going to drop or which sperm is going to be put into the equation, we have to take that into account, and we have to think about this as a probability. We have to say that there's going to be a 50% chance, a 1 in 2 chance, that this egg actually has the big R in it, okay? Because there's also a 50% chance that it could have the little r, okay? So let's go about it with that in mind. Okay, we think to ourselves, all right, let's look at it and say, if we want this to happen, if we want big R, little r, or pardon me, big R, we have to have the other one of these be a big R from mom. And so there's a 50% chance that this actually occurs, okay? Now, when we take this into account, okay, we now have to figure out the overall probability that this is going to occur, that this event is going to occur. We want this big R right here to be with this big R at the same time. So the way we can say it is we can say we want this and this to happen in the same baby. Okay? And so to figure this out, we have to multiply the individual probabilities that this might occur. Okay? So we say, all right, 1/2 is the individual probability that dad is going to give his big R. 1/2 is the individual probability that mom is going to give her big R. So we have 1/2 times 1/2 equals 1/4. Okay? And so I know that I use that one just because it was in the upper left-hand corner, and that's how I read this stuff. But the same thing applies to the actual question that's been asked. Okay? It says, what is the probability that given these two parents, okay, right here, this dad right here, and this mom right here, given those two parents, that we get a homozygous recessive individual. Okay? And so the same rules are going to apply. Let's look at this one too. The same thing. We know that to get this baby, we have to have two small r's. We know that one of those small r's has to come from dad, and the other one has to come from mom. Okay? That's how the babies are formed. So let's take all this into account. Now, what is the chance that this sperm is actually going to have a little r in it that we need? It's 50%, right? So if we need this little r, there's a 50% chance that we're going to get it. Okay? A 1 in 2 chance we're going to get it, because the other chance, the other possibility is that it's a big R. All right? And then if we want this little r from mom to fulfill our homozygous recessive individual, there's a 50% chance that this egg is going to have a little r in it. 50% chance right there. Okay? Because the other chance, the other possibility is that it could be the big R. So if we want the small r from dad and the small r from mom in the same baby, which that's what we see here in this particular cell, we would have to multiply the individual probabilities of those events. So that would mean we would multiply this individual probability with this individual probability. So we would have 1/2 times 1/2 equals 1/4. Okay? So the reason I've left this Punnett square up here is because you know Punnett squares. You guys know how to do Punnett squares, which is great. You should, because they will help you. But it also helps to verify the answers. You know that if I'm looking to figure out, okay, I've got heterozygous parents, big R, little r, that if I were to just run a normal Punnett square and I wanted to figure out what's the possibility that I get a homozygous recessive baby, you could look at that Punnett square and say one of these 4 boxes Okay, this one here, one of these 4 boxes had the little r, right? And that would make sense to you. But I also want you to verify that it's the same as what we just calculated right here, okay? Because we have calculated the individual probability of getting this little r and the individual probability of getting this little r in the same baby, and that is where we use the rule of multiplication. We want this and this to happen in the same baby, okay? And when we do that, we multiply, okay? So that is the rule of multiplication. I personally think the rule of multiplication is the easier of the two to use. It doesn't necessarily mean that we can't use the rule of addition, because we will. But most oftentimes I think we will probably use this rule of multiplication. Okay, so let's talk about the addition rule. Okay, the addition rule of multiplication, the addition rule of probability states that events that can occur in 2 or more different ways will occur is the sum of the separate problems. Okay, that's kind of a mouthful, and it's weird, it's odd to say that, right? So another way of looking at this, another way we decide whether or not we're going to add instead of multiply, is to say that if I'm happy with this baby or this baby, I would then add the probability of this one occurring to the probability of this one occurring, and that would give you an overall probability of either one of these 2 babies happening, okay? Because we'd be happy with either one, okay? So this one I think is more oftentimes easier. It's easier to see what's going on when I walk through the next diagram, okay? Now, the question that you see here, in which I will walk through it in the next one, and I'll try to remember again a little bit sooner, but in a cross between pea plants that are heterozygous for flower color, so we've got the exact same parents as we did in the previous one. We want to know, what is the probability of the offspring being heterozygous? Okay? We want to know what's the probability of the offspring being heterozygous. And it's a little bit different process to go through. Now, one thing I want you to kind of keep in mind here is that when we go through these, let me go ahead and snapshot this. When we go through these, this example, specifically the addition rule of probability, oftentimes you're going to use the rule of multiplication as well. Okay, so that's one of the reasons I went through the rule of multiplication first, is because a lot of times you use it to solve these addition problems too. Okay, now this one's got some fancy highlighting. Now what we hold on, let me go ahead and bring the highlighting up. Okay, and then I'll take a picture. Okay, that's an ill-placed laser dot. Hold on. That's not going to work either. How about up here? There we go. All right. So,
Williams, please report to the main office. Michelle Williams, please report to the main office.
all right. So this one is similar in nature, except for what we're trying to find is the probability that we get a heterozygous baby. Okay, a heterozygous baby. So let's go ahead and start calculating the probabilities that we get a heterozygous baby at all. Okay, so if we're going to start out with, again, heterozygous parents, big R, little r for the dad, and big R, little r for the mom, and we're going to segregate those alleles into separate egg or sperm, kind of the same way we see previously. Okay, and then those egg and sperm are going to combine to make a baby, right? Now, when they combine, we again don't know, are they going to get this one or are they going to get this one? So we have to take into account that there's probabilities there. It's a flip of a coin. It's a 50-50 shot whether or not the baby gets the big R or the little r. Okay. Now let's see what the possibility that we get a heterozygous baby from these particular parents. Let's look at this case first. Okay, upper right-hand corner one. Because we notice that in this part of the Punnett square, there's a big R and a little r. Okay? Now, let's go ahead and figure out where these r's happen to be coming from, because I think that can help us understand a little bit of what's going on as well, and where the r's actually are coming from. Now, you guys know how the Punnett squares work, right? You know how the Punnett squares work, where you take this r and you kind of bring it all the way over, right? Come on now. You'd take this r and bring it all the way over, and it would be this guy right here. And you'd take this little r right here, and you'd bring it down, and it would be this guy right here. Right? That's how normal Punnett squares work. Now, let's take a look at the probabilities that this happens. Right? There's a 1 in 2 possibility that this egg with the big R actually goes into this particular baby, because we don't know for sure if it's going to be the big R or the little r down below. But we know that there's a possibility that it is the big R, and that possibility is 1 in 2. Okay? So we have a 1 in 2 chance of this event happening right here. And we also have a 1 in 2 chance that this event happens right here, where this little r is going to be in the baby. Okay? So there's a 1 in 2 chance of that event happening as well. So this is where we'll start to apply our rules of multiplication. We want to know what's the possibility, the overall possibility that this baby right here that we just made right there is actually going to be born. Okay? So the way we have to approach this, we got to say, I want this r and this r together in the same baby. Okay, so we got to multiply the individual probabilities, right? So we take a 1/2 times 1/2 equals 1/4. So there's a 1 in 4 chance that this baby is born. Okay, that this particular baby in this box is born. There's a 1 in 4 chance. We verified it 2 different ways. We verified it by making a Punnett square, which you see, and we also verified it using the multiplication rule of probability. Okay? You said, Sutherland, we're doing the addition rule in this slide, you moron. I know that. So we're going to get to the part where we do the addition part in a second. Okay? Because what we notice is that there's actually a secondary way that we could possibly get a heterozygote, right, for a baby. Now, the difference is which parent is giving the allele. Okay, so let's look at it. We'll say, look, all right, this right here is a big R from Dad, and we would bring that one down, and it would go into the baby right there. Okay, that's what we see here. And there's a little r that would be brought over, and it would go into the baby right here. Okay. So now again, we aren't sure if this sperm is going to have a big R or little r, so we have to put a probability on it. Okay, we're going to say this probability of this big R happening or being in that particular sperm that goes here is a 1 in 2. Okay, so there's a 1 in 2 chance that this big R is put into that baby. And there's a 1 in 2 chance that this little r right here is put into that baby right here. 1 in 2 chance. Okay, so we want this R and this R together in the same baby right here. So what we're going to do is multiply the individual probabilities. So 1/2 for the big R times 1/2 for the little r equals a 1 in 4. And again, we can verify this. We just verified this using the rule of multiplication because we want this r and this r in the same baby together. And we've also drawn the Punnett square that shows it, right? One of these 4 boxes is this baby right here. So we've got a 1 in 4 chance there. Now for the application of the addition rule. Now we can say, if I'd be happy, let me get a different color pen out here first. I would be happy with this baby right here or this baby. Then I will use the addition rule. I'd be happy with either of those babies. That's a great situation for me. I like this one here or I like this one here. So then we have to take into account the individual probabilities of each of these events that I just circled in green. And we just verified that the individual probability of this event up top is 1 in 4. And the individual of this event down here in the lower left is 1 in 4. we then add those individual probabilities together, because we said I'd be happy with this 1/4 or this 1/4. So we add those individual probabilities together and we get the final answer. Get the final answer of 1/2. Okay? And again, you can verify that by doing Punnett squares. 2 of these 4 boxes result in the heterozygote baby that you want. And that's a 1/2 chance, right? So it can be complicated. The more you use them, the more you apply these 2 rules, the easier it becomes. Okay, and so that's one of the things that we're going to do. But one of the things that I want to express to you here is that when you do, I'm taking you on a tour of the room here. When you do certain types of problems, let's say this is difficult here, like this is a trihybrid Punnett square. It's a trihybrid Punnett square. And the trihybrid Punnett square has 64 boxes. That's just 3 traits. Okay, that's 1 more trait than what you would do for a dihybrid, but it exponentially increases the amount of boxes you have, right? Instead of 16, you've got 64. Okay, and it's much more complicated. Imagine what that would be like if you did 5 traits or 4 traits or 15 traits. Okay, very, very difficult stuff. But with these particular probability rules in hand, you can do any of those types of problems much, much more easily if you know exactly what you're looking for. Okay, so that'll do it for the crash course on the use of the probability rules of multiplication in genetics problems. The next thing that we're going to do is to have some of those trihybrid crosses or those tetrahybrid crosses and see how we can apply the rules that you learned in this video. All right, I'll see you then, guys.